start. So IgG, in addition to IgA, it can bind and neutralize the toxins. And another important property is opsonization. So here what happens, this IgG, it coats the pathogens and prevents them from entering into the host cell. Okay, and the other properties, we have already seen this. And this Fc receptors, it also mediate the antibody dependent cellular toxicity. First, what happens is immunoglobulin, it binds the antigen surface on the target cells. Then the Fc receptors on the natural killer cells, they bind to the Fc portion of the immunoglobulin. Then there will be cross linking of the Fc receptor signals to the natural killer cells to kill the target cell. So this target cell, it dies by apoptosis. Okay. So here the target cell is opsonized with the antibodies and thereby targeted for destruction by the NK cells. So this is about IgG. Then coming to IgA. So it is the second most abundant class and it constitutes about 10% of the serum immunoglobulin, and the half-life of it is 6 to 8 days. And it is a major immunoglobulin which is found in the secretions like saliva, tears and colostrum. So this IgA, it has two forms. One is serum IgA and the other is secretory IgA. Serum IgA, it is monomer, and the IgA on mucosa or secretory IgA, it is a dimer. Okay. And here we have a J chain in IgA. So these J chain, it facilitates the transport of IgA across the mucosal epithelium, and it also facilitates the transfer of IgA to the newborns to confer the neonatal passive immunity. So then now coming to secretory IgA, it is concentrated in secretions mostly and also on the mucosal surfaces. So it helps mostly in the local immunity against intestinal pathogens and also respiratory pathogens. So it is very resistant to the digestive enzymes. It inhibits the adherence of organism to mucosa, thereby it activates the alternate complement pathway. So what is this alternate complement pathway? You already know, but still if we are taking any revision classes, we can go for this pathways. Okay, and it also promotes phagocytosis and intracellular killing of the microorganisms. So this is about IgA. Coming to IgM, it constitutes only about 5% of the serum immunoglobulin and the half-life is 5 days. So it is a heavy molecule, that's why it is also called as millionaire molecule. Remember IgM, actually it denotes mu, Greek letter mu, but you can remember it as M for millionaire also. So it is a millionaire molecule because it is a heavy molecule and it is a polymer. It has 5 peptide subunits. Okay, and this polymerization, it depends again on this J chain. Okay, so most IgMs, these are intravascular, and remember this IgM, it is the earliest immunoglobulin to be synthesized in the fetus or newborn. And this IgM are also very short lived and they disappear earlier than IgG. So here we can see this IgM, it exists as a pentamers and it has 10 different antigen binding sites. Okay, so it has very high avidity. So as with immunoglobulin A, it has also a J chain for secretion. It is the first antibody expressed in the mature B cells. So it is again important for the activation of the complement pathway. It appears early after infection and it's usually not seen upon reinfection. Okay. Then coming to IgD. So this IgD structure is very similar to IgG, and the concentration is approximately 3 milligram per 100 ml of the serum, and it is again intravascular. So its function is the recognition receptor for antigen. Okay, and it is present on the surface of the unstimulated B lymphocyte. Coming to IgE. Again, the structure of IgE is similar to that of IgG. It has very much affinity for the surface of tissue cells like mast cells. It mediates PK reaction, that is Prosnitz-Kushner reaction. And very high levels or elevated levels of IgE are seen in asthma, hay fever and eczema. So this IgE, these are produced in the lining of respiratory and also intestinal tract. So IgE, it is involved in the mast cell activation. So this IgE, it binds the mast cells where it waits for the antigen. And this IgE, it is different from IgG, which must bind antigen before the cells. So when an antigen cross-links IgE, it causes mast cell degranulation. So immunoglobulins are normal. Then what are these abnormal immunoglobulins, where we see few conditions like multiple myeloma, Waldenstrom's macroglobulinemia, heavy chain disease, cryoglobulinemia. So Waldenstrom's macroglobulinemia, it is a cancer of mature plasma cells which causes overproduction of IgM. And in heavy chain disease, there will be overproduction of FC, parts of immunoglobulin heavy chains. And in cryoglobulins, these are single or mixed immunoglobulins that undergoes reversible precipitation even at low temperatures. It results in this condition of cryoglobulins. Then coming to antibody diversity. So this immunoglobulins are subdivided into three types: isotypes, idiotypes and allotypes. Okay. So isotypes means here there is antigenic difference in the C region. 5 different immunoglobulin classes are different serotypes. Then coming to the allotype, here antigenic feature of the immunoglobulin that may vary among the individual. It is mostly under the genetic control or because of the antigenic difference in the C region of H chain and L chain. Okay. Then coming to idiotype means there will be antigenic difference in the V region, that is variable region of the immunoglobulin. That is about idiotype. So why there is antibody diversity? Because of presence of large number of antibodies that bind different antigens or pathogens. And the significance of this is to develop engineered antibodies for therapeutic purpose. And here in antibodies, you should learn an important term called class switching. Okay. So here what happens on antigenic stimulation of any particle, the H chain, that is the VDJ unit, the H chain VDJ unit, it can join any constant heavy chain segment, and it expresses the particular antibody class. So this process is called class switching. Okay, this is mostly seen in hyper IgM syndromes. Okay, then coming to next another subtopic in antibody, you should know about primary antibody response and secondary antibody response. Primary antibody response means, as the name itself suggests, it is the first exposure to the antigen. Secondary antibody response, it is a subsequent exposure, means any exposure after the first exposure. And the lag period for primary antibody response is days or few weeks, whereas for secondary antibody it is few hours. Here large amount of immunoglobulin is present, whereas in primary antibody response there is smaller amount of immunoglobulin. So here in primary antibody response, antibody increases in weeks then decline, whereas in secondary antibody response they persist for long periods, and the antibody responsible for this is IgG, whereas for primary antibody response it is IgM. Then now coming to monoclonal antibodies, it is a very important question. Okay, so monoclonal antibodies means, as the name itself suggests, antibodies which are derived from a single clone, antibodies which are derived from a single clone of plasma cells, all having the same antigen specificity. That means, that is, they are produced against a single epitope of an antigen. So this concept of monoclonal antibodies was first discovered by Kohler and Milstein, and they were awarded Nobel Prize. So the monoclonal antibodies, it is based on these three principles, that is combination of plasma cells and myeloma cells which result in hybrid cells which can produce antibody of required specificity. So very briefly we will see about this technique of monoclonal antibodies. So here the mouse splenic cells, mouse splenic B cells, mouse is first injected with an antigen which contains the desired epitope. So after an interval, what happens? These mouse splenic B cells are obtained, which contains mixture of B cells which are activated against the epitope of an antigen injected. And myeloma cells, these are immortal cells, okay, and these are cancerous plasma cells which closely resemble mouse B cells. So these are compatible for fusion. So that's why these myeloma cells are taken, and they have the capacity to produce their own antibodies. So these myeloma cells are genetically modified with two mutations, that is double mutation. So that why double mutation has to occur? So that they lose their ability to produce their own antibody, but they still retain their immortal property. So what happens is, myeloma cells and splenic B cells of the mouse are fused in PEG, that is polyethylene glycol broth. So in this reaction chamber, three types of cells are generated. Here you can see three types of cells. One is unfused myeloma cells, these are unfused myeloma cells, unfused mouse splenic B cells, and fused hybridoma cells. So how is this carried? This is carried out by subculturing the cells in a reaction chamber on a special medium called as HAT. What is HAT? Hypoxanthine aminopterin thymidine medium. Okay. So this aminopterin, it blocks the de novo pathway so that the cell has to perform the salvage pathway. Why? To synthesize the purines for its survival. Okay, I am not going into the detail of those pathways. So what happens? What is the fate of these three cells which are formed on HAT medium? The unfused splenic B cells, they can grow, but they do not survive long as they are not immortal. And the unfused myeloma cells, they cannot grow as they lack this HGPRT enzyme to perform the salvage pathway of purine synthesis. And these hybridoma cells, they can grow and they survive long. Okay, so these hybridoma cells are taken for further process. So if the original antigen used has multiple epitopes, then many B cells would fuse with myeloma cells to produce a mixture of hybridoma cells. Then this medium which contains hybridoma cells is then diluted into multiple well plates to an extent that each well contains only one cell. Okay. Then this hybridoma cells, how are they maintained? One is these can be cultured to generate a clone of identical cells, means they produce pure form of monoclonal antibodies. And another way is by the desired hybridoma cell, it is injected into the peritoneal cavity of the mouse where it can multiply and produce monoclonal antibodies in the ascitic fluid of the mouse. So such monoclonal antibodies which are obtained from the mouse and serum may not be in pure form, means they might be mixed with another antibodies. So how are they purified? They are purified by immunoprecipitations or by any chromatographic techniques. Okay, so this is about the explanation of monoclonal antibodies. And the clinical use of this antibodies is, it is used as diagnostic, imaging and therapeutic reagents, and it detects pregnancy, and it helps in diagnosing various pathogenic organisms, and it is used to measure the blood levels of various drugs, matching histocompatibility antigens and detecting antigens shed by certain proteins, and it also acts as immunotoxins. Okay, so this is about monoclonal antibodies. Now this is a homework for you. Okay, so wherever there is a question mark, you fill this table, okay, and see whether it is correct or not. Anyways, I am showing you the answer also, but don't jump onto the slide of an answer. Okay, first try to fill this, then see the answers later on. Okay, so with this we end our division class of antibody. Okay, don't see the answer slide next to this.